meeting. And thanks to the Clinical Sciences group for giving me the opportunity to present the solutions and products that Calixa offer to obtain native membrane proteins suitable for drug discovery. So today I will speak about our platform and present you one of our protein targets, A2AR, and the work achieved with this native protein. Membrane proteins represent more than 60% of all therapeutic targets, including G protein-coupled receptors, ion channels, and transporters. However, it is a real challenge to study them, and as for today, only 2% of the 3D structures found in the PDB are membrane protein structure. The reasons are the difficulty to produce and isolate the membrane targets, which comprise an hydrophobic domain, and the conventional processes of extraction that affect the native state and function of these targets, all resulting in...
Alexis, sorry, apparently there is a problem with the presentation. Could you please stop sharing and, oh no, it's okay now. Oh, so, you just have to share again your presentation, sorry.
Okay, I will share again my screen. Okay, so is it okay? Okay. All these difficulties leading to an impact on the development of drugs, antibodies, vaccines, and diagnostic applications. This is where Calixa comes in to propose a native isolation approach. In collaboration with Grégory Durand, S2CB chemistry team at Avignon Université, we work as a joint laboratory named Chem2Sab to develop proprietary molecules that act as non-denaturing detergents. These molecules are used to purify native membrane proteins without the need of refolding or mutagenesis, and greatly improve protein stability and purity while maintaining their functional and structural integrities. We offer 4 different families of surfactants with various amphiphilic properties, allowing more diversity to membrane protein extraction and stabilization. It includes calixarenes, diglucose, lactobionamides, and tris-acrylamidomethane derivatives, all available individually on the Clinical Sciences website, and soon as a screening kit. In addition, we are currently developing SMA polymers for the extraction of proteins as nanodiscs in their native lipidic environment. Calixarenes are made of a polar ionic crown and an alkyl chain. The alkyl chain allows detergent-like properties, and the grafting of glucose to the polar head can improve its stabilizing properties when used as an additive. We also developed a soluble cholesterol derivative that mimics cholesterol and stabilizes eukaryotic proteins. Diglucose surfactants are made of a branched non-ionic polar head group and an alkyl chain that make it soft detergents not sensitive to ionic strength or pH variations. Lactobionamide contains a linear non-ionic polar head and an hydrophobic chain that comes into 2 flavors, either hydrogenated or fluorinated. And finally, the TAC family is made of bulky polytris polar heads and also comes with an either hydrogenated and fluorinated chain. These molecules help us purifying native full-length membrane proteins that you can purchase online through Clinical Science. And we propose different therapeutic targets, including the human GPCRs, adenosine A2A receptor, and melatonin receptor involved in the central nervous system and in the success of therapies against many diseases, such as Parkinson's disease, depression, and even cancer. We also offer bacterial transporters involved in antibiotic and multi-drug resistance. So this is ACRB and BMRA. And more proteins are coming soon, including the muscarinic receptor 4, the MRP4 transporter, the KCC2 ion channel to find treatments against pain and epilepsy disorders. We also work on the chikungunya spike protein, which is currently investigated for vaccine development, and the B lymphocyte antigen CD20, which is a clinical target in the treatment of lymphomas, leukemias, and autoimmune diseases. I will now go more into details on our A2A target. So the adenosine A2A receptor is found in platelets in the brain, cardiovascular system, and in activated T cells. It acts as an allosteric modulator of dopamine receptor and is involved in vasodilation, inflammation protection, and tumor growth regulation. Because of its wide repartition and roles, it is a main target for drug development against encephalopathy, Parkinson's disease, insomnia, pain, depression, and hypertension. To study it, we produced the full-length wild-type protein as a recombinant protein with streptavidin and polyhistidine tags cleavable by TEP protease. The protein was produced in insect cells, from which we isolated the membranes that we subjected to solubilization. So here you can see that whereas the DDM-CHS combination commonly used for GPCR fully solubilized the protein here in the soluble fraction, we achieved full solubilization thanks to our calixarin derivative. And because DDM-CHS was described in the literature as suitable for A2A purification, we used it in combination with our calix. The protein was then purified by IMAC in phosphate saline buffer, complemented with a minimal amount of detergent. The protein was highly pure with very little degradation. Its functionality was then validated by a radio binding assay, proving that the protein binds both its known CGS agonist and its high affinity ZM inverse agonist in the nanomolar range. Moreover, the purified protein was found to be highly stable without any sign of aggregation after 7 days at room temperature, as observed by steric exclusion chromatography. And the protein thermostability here in dark blue was greatly improved in comparison with the purification in DDM-CHS only. And it was as stable as the STAR2 truncated and mutated thermostabilized version of A2A. Here, the use of our detergent avoids the need to introduce any mutation to stabilize the native functional full-length protein. After validating the quality of the protein, we use this as the proof of concept that Calixar's approach is compatible with drug development in a collaborative work with the Université de Lyon, Université de Paris, and Institut Cochin first. And we proved that our protein was compatible with small molecule screening by STD-NMR. So controls were performed where we could observe the binding of caffeine, which is an antagonist of A2A, and the binding of CGS agonist. We could even perform a competition assay that showed that bound caffeine is displaced by the addition of ZM antagonist, allowing us to conclude that both drugs bind to the same pocket. Then we screened for drug fragments binding and identified 10 molecules that displayed weak or strong affinity for A2A. Among them, none acted as agonist of A2A in terms of CAMP production, but 6 were identified as antagonists of the protein. Our protein was also successfully used by Edelris for high throughput screening by affinity selection mass spectrometry, and it allowed also to the identification of several drug targets. Deuterium exchange mass spectrometry was also performed at the Université de Strasbourg and allowed the detection of residues interacting with adenosine and with CDN within A2A. So with this technique, we had a good coverage of the protein, and we can see the critical residues of the protein that interact with the drug, which allow drug design and more drug development. Finally, mice were immunized with either our protein or the STAR2 truncated version. And serum antibodies were analyzed by ELISA. And we showed that our purified protein induced higher immunogenicity than the mutant protein against native A2A from membranes or from the purified protein. So in addition to the catalog proteins, we also offer on-demand production, starting from the gene and expression of the protein to its purification and quality controls, which allowed the client to develop its own drug discovery clinical assays. And over the years, our proteins allowed the study of several therapeutic targets. We also recently published an application note in collaboration with Nanoimaging Services, in which we solved the 3D structure of ACRB trimer, which was purified exclusively with a calixarine detergent. So this time we didn't use any DGM or any other combination of detergent. It was calixarine only. And it maintained the trimer assembly of the protein and allowed us to solve the structure, which makes it compatible with cryo-EM. Okay, so for more details and requests, feel free to visit Clinisciences and Calixar websites. And I will now be happy to answer your questions. Thank you all for your attention.
If you have any question, you can ask your question now. You can activate your microphone, or you can use the chat to ask your question.
Hi. Yeah, my name is Toby. It was quite a good presentation. I was going to ask, have you tried to express these proteins in mammalian cells before?
Hi, Toby. Thanks for your question. We didn't, because we knew that the GPCRs, in case of A2A, were nicely expressed in insect cells. We did try in Pichia, which is yeast, but we did not try in mammalian cells. So nowadays, our incoming new targets are expressed in HK cells. So yeah, now we are switching to mammalian cells, but okay, we didn't with A2A.
All right, because I was just wondering about PTMs that are associated with mammalian cells, and the proteins being really functional with the various PTMs?
Yeah. That's a good question. And A2A has glycosylation, and we checked that it has this glycosylation, but there are still slight differences between the insect PTM and mammalian PTM, but still the protein was active. So we were happy with that.
All right, thank you very much.
Hello, it's me, Roshan here. So I have a general question on what's the principle behind how you select the detergent just for any membrane protein actually? Is it just a hit and trial iterative process, or is there some physicochemical principle that can help in determining what detergent to use?
Yeah, it's actually very protein dependent. We can aim at a detergent family if we know the protein families like GPCR. They work well with some of our calixarin, but yeah, unfortunately, sometimes you just have to screen them to get the best detergents.
The best would be to get the membrane and then screen it. That's the best approach, right?
Yeah.
Okay. Thank you.
We have a question in the chat. Do you have any suggestions for extraction of cell wall proteins from yeast detergents?
Okay, so this is a good question because we didn't work much on cell wall proteins from yeast. We did extract membrane proteins from yeast, but normally, if you can isolate the membranes and try a different detergent, you should be able to solubilize the proteins. Yeah. What kind of detergent you used? So yeah, we used our detergent. So the one you saw, and more in particular the calixarin detergent at that time with the calixarin chrome. Okay, sorry. So we use the calixarin derivatives. So it's the molecules with the calixarin chrome, and it worked. Yeah, this one. It worked at some point with yeast, but we still had better extraction in insect cells, but it worked. Have you tested your detergents or another methods for bacterial membrane proteins? So yeah, yeah, of course. So we used it on ACRP and DMRA, which are bacterial membrane proteins. We produce it from E. coli, and the structure of ACRB that we got here. To determine the structure, we solubilize the protein directly from E. coli membranes with the calixarins, and we kept the calixarin during the purification process. We just lowered the quantity of detergent during the purification, but it was the same detergent all along, starting from membrane solubilization of E. coli to the purified protein on a cryo-EM grid. So yeah, it works well.
There is another question, but maybe you answer. Have you tested your detergents or another methods for bacterial membrane proteins?
Yeah, I just answered that.
Other questions? Oh, there is another question. Do you have a standardized purification protocol for the detergents? Because all of the downstream steps are crucial for proteins quality and stability.
Oh yeah, okay. So yeah, we usually prepare membranes at a final concentration of 5 milligram membrane protein per milliliter. And we use our detergent at a concentration over 10 CMC. So this is an important factor because if you use too much detergent, you will have trouble in purifying the protein. And if you use too low detergent, you will not extract the protein correctly. And we also, so in terms of buffer, we usually use PBS like buffer, so phosphate buffer with 150 millimolar salt. And well, that's it. And we keep everything at 4 degrees. We take care of that. So. Oh yeah, and we only 2 hours solubilization work well with our detergent. So we usually do 2 hours solubilization with agitation in the fridge. I hope this answers your question. Thanks.
Other questions? Oh, yes. What type of CMA are you proposing to develop? What will be their advantage, inconvenience in comparison to ones available currently?
Yes, that's a good question. So I cannot fully answer that because of confidentiality, but we are currently working on a purification synthesis method of regular SMA. So the one that you can find commercially, that is say the SMA 2:1 and 3:1, but with a more controlled synthesis process, because most of the batches that are available are different from a batches to another, and sometimes it's not really pure with some additions of malic acids between the polymers. So we work on a more controlled process, and we also work on another design of polymers, but I cannot say too much about that yet, unfortunately. And the advantage is, so it will be, for the regular SMA, it will be a more controlled, more stable production of polymers and purer molecules. And for the incoming molecules, we will try to produce polymers that are not subjected to aggregation depending on the pH or to absorbance in the UV. And also salt tolerant, because this is a huge trouble with regular SMA. Any other question?
Okay, so if there is no other question, we can stop here. Don't hesitate to send us your questions. So you can send your question to Clinisciences, and we will discuss with Calixar in order to send you the answers. We record this webinar, so it will be available on our website, I think tomorrow. So thanks a lot for your attending to this webinar. Thanks a lot, Alexis, for your presentation. And
Thank you very much.
have a nice day, everyone. Thank you.
Have a nice day.
Bye-bye.